Hi, everyone. My name is Riccardo. I'm one of the co-founders at Alithea. Today, I'm going to be talking about a new technology that we have launched since now a couple of years, and that we have seen as really being used widely. A couple of things about Alithea. Alithea is an EPFL spin-off, so we are a Swiss company. We're now a Swiss-American company. Actually, we have two sites, one in Switzerland and one in Maryland in the US, where we actually do distribution and our services as well. Alithea is focused on the development of new tools for the generation of transcriptomic data. We are one of the companies that you may say is helping to improve the way we develop new drugs, the way we actually develop and screen new compounds. And we're taking the approach of the point of view of data generation. Of course, there are other dimensions like improving the model systems that we use and the way we analyze the data, we integrate the data. And the overall overarching goal is to really be able to take decisions earlier in the pipeline, to be able to actually understand the biology of the system and what the chemicals that we are studying do to the system earlier than that was possible before. Conceptually, what we typically look at is transform the typical V-shape drug development funnel into the T-shape, in which we basically take decisions earlier on to really save money and time down the road. As I was mentioning, Alithea is focused on one of these aspects, which is data generation and specifically transcriptomics. So we are hyper-focused on high-throughput transcriptomics, and the technology that I wanted to present today is really summarizing and fitting well in this dimension. DRUG-seq is a technology that we developed to be offering the best of both worlds. The two worlds being one, targeted assays, in which you actually have to predetermine what you're measuring, like microarrays. And the idea of these ones was high throughput and very low cost, but then it offers a disadvantage, which is the fact that you have to know a priori a little bit about the biology of the model that you're studying, and the assay is, per definition in this case, biased. The other side is the RNA sequencing side. RNA-seq is, of course, a very interesting and exciting technology because it allows you to actually measure everything that is expressed in an unbiased manner, but current implementations are, of course, very expensive and low throughput. The idea of DRUG-seq, especially the DRUG-seq version that we have developed at Alithea, is to really offer a solution to these trade-offs and offer a solution that is extremely high throughput. It's ultra scalable in terms of cost, so we're really optimized the procedures, the reagents. Some of the reagents we actually produce them in-house as well to be able to offer extremely scalable costs. And by scalable, I mean that the more it's done with DRUG-seq, the lower the unit cost, the per sample cost actually gets. And the ultimate advantage is that it's an unbiased approach. So you can actually measure everything that is expressed on all the mRNA transcripts without actually having to predetermine what you're measuring. Just to give you really a brief overview of DRUG-seq in a nutshell. DRUG-seq typically starts with frozen cell plates. DRUG-seq is compatible with 9634 and 1536 well plates. And the first key aspect of DRUG-seq is that it's an isolation-free procedure. So there's no need to isolate RNA through any type of magnetic bead or column purification. The way the protocol starts is by basically just adding a lysis buffer that we offer on top of the frozen cells. This basically solubilizes the RNA into solution, which then can be basically directly implemented in the subsequent steps. And the second key aspect of DRUG-seq is that the procedure is massively multiplexed, meaning that we can actually tag at the very beginning all the samples in a sample-specific manner, after which we can basically pool all the samples in a single tube and process them in a tube for the rest of the workflow. This drastically decreases the amount of reagents and time and tips that actually need to be used to run the workflow. And this simplicity is actually what makes it unique in the sense that it's the only RNA-seq workflow in our knowledge that can actually be implemented at the scale of, for example, 1536 well plates. It's very compatible with all kinds of next-generation sequencing solutions. This has been validated on Ultima, Element, in Illumina, and even MGI. And the data analysis is, of course, open source. There's nothing proprietary in the way we analyze the data or we provide the data. Everything is open source, and we typically also provide support with the data analysis as well. DRUG-seq can be accessed in different formats. For those that actually have capacity in-house in terms of NGS and library prep, we offer kits with all the reagents needed to go from cells to sequencing-ready libraries. We offer end-to-end services as well, in which you basically just need to receive the frozen plates, and we take care of everything from library prep, sequencing, and data analysis. And recently, we also started partnering with CROs to actually offer an end-to-end solution to really accommodate also the cell biology and treatment part. Here, I just wanted to show a couple of case studies of projects that we have run in the past that we're actually allowed to discuss. Just to give you an idea of what's possible with DRUG-seq and what type of analysis and data can be generated from this. This was one actually of the first DRUG-seq projects that we have run in collaboration with the Broad Institute. This was an interesting project because they were studying compound combinations. So we actually profiled over 3,800 compound combinations for a total of around 11,000 samples. We detected around 12,000 genes per sample. And this was actually a very fast project that actually took only 7 days from the moment we received the plates to the moment the data was delivered. Here's just to give you an idea also about what type of analysis typically is possible with DRUG-seq data. The data is, of course, very abundant. Even if it's bulk RNA sequencing data, it starts to look more and more like single-cell RNA-seq data. So typically what we see as the first layer of analysis is actually clustering approaches like UMAP, PCA, TSNE, and marker gene identification. And then typically, what one can do is to actually isolate a subcluster or a few clusters of interest, and then zoom in and understand what's happening actually there in that cluster. Specifically for this project, the idea was to actually understand what compound combinations were doing, how they were interacting, and what compound combinations were actually bringing back the gene expression state to the baseline. So specifically for this project, we actually had to do thousands and thousands of pairs-wise analysis, which is not typically done if you're not looking into compound combinations. Another interesting project that we have done in collaboration with our partners at Axam, the number of samples was actually bigger in this case. And it was interesting because here we actually did DRUG-seq on several cell lines without any compatibility issue. So DRUG-seq is very compatible with different cell lines. We do not need to optimize the procedure or the lysis step for every different cell line. It typically works right off the bat with different cell lines. The idea here was to understand and get early views on markers for toxicity for the compounds that were being processed. And in this case, we started looking at specifically clusters, how the compounds were clustering, and whether there was a compound-specific or a cell-type-specific clustering effect. And you see how the data actually can be quite sparse when you start having such a large data set. Specifically here, we started seeing a compound-specific effect. You see that actually the samples are actually clustering out with higher dosage of the compounds. And again, one example of the analysis that we have done in this case was to actually check what compounds and what cell lines were actually reacting to different compounds. So here in this case, for example, we started looking for specific signs of mechanism of action for these cell lines. You see here, for example, in this specific cluster, we could actually identify a compound that was actually setting out this cluster at any dosage, so we didn't see any specific dose-related response, and we saw how this compound was actually acting differently based on different cell lines, whereas this compound was actually acting more gradually, so it was actually giving a more dose response to the cell lines. DRUG-seq is one of those technologies that require a conscious shift in the way the investments are done in drug discovery. It's an investment that is really done with the view of increasing the amount of data that you can actually digest and play around with at the early stages of drug discovery. And this is the type of data that we see can really bring a benefit down the road, because it allows you to take decisions faster, and it can bring significant advantages like identifying liabilities early, accelerating go-no-go decisions, smarter target selection, and also you can actually export and extend the knowledge that you have accumulated early in the discovery phase to actually clinical trials, which can bring, despite the relatively small investments at the earlier phase, much faster decisions and effective decisions down the road. Here, I just wanted to mention also a new technology that we have just developed. We're about to launch it actually in July, and it's a new system. It's, I would say, a big bet of Alithea's, and we're looking for partners at the moment that are interested in the extra layer of information that this technology will bring. The technology is called Total DRUG-seq, and the idea in this case is that Total DRUG-seq brings the same advantages as DRUG-seq. It's extremely high throughput, it's scalable and low cost, but in addition to 3' RNA sequencing data, in addition to gene expression data, we can actually profile long non-coding RNAs as well with full-length level information. So we can actually sequence everything across the RNA molecules. Here, just to show that Total DRUG-seq is indeed a full-length approach. Typically, Total DRUG-seq was compared with standard non-multiplexed and non-extraction-free approaches like NAB. And as you can see here, Total DRUG-seq really, in a way, profiles the entire length of the RNA molecule. As I was mentioning, Total DRUG-seq brings an additional layer of information. So we actually, in terms of genes detected, we are actually at par with DRUG-seq, so we can actually detect the same number of genes. But when we start looking at specific transcripts, we can detect significantly more transcripts, so we can actually start doing transcript-level analysis. And the partnerships actually we're looking for at the moment is to understand how important this additional layer of information can be in the discovery phase or any analysis that might be interesting to do with Total DRUG-seq. One example of an internal analysis that we started doing was to understand whether these data could provide insight into alternative promoter activation. Here we considered a model in which U7 cells were treated with different doses of TGF-beta. And we started looking at, we basically profiled these cells with standard DRUG-seq here in green and Total DRUG-seq in different shades of blue. You can see here that with standard DRUG-seq, the only thing that we can say here really is that the gene is actually expressed, which is perfectly fine when we're looking specifically at differential gene expression analysis. But if we didn't do Total DRUG-seq in this case, we basically would be missing out on a potentially interesting biological phenomenon, which is this alternative promoter activation. Here you see that basically promoter number 1 is activated with lower or no TGF-beta doses, whereas with a gradual increase in the dosage, this was actually profiled on 96 different samples, you see that the second promoter gets activated. And we found it interesting because promoter 2 is mark of cancer and tumor progression. And this was, again, a type of biology that would not be identifiable if someone was actually looking only specifically at 3' RNA sequencing data. So this is, just to give you an example of analysis that we started doing. If this sounds interesting, if this is the type of data that you would like to explore, please reach out, because we are very interested in partnering and actually looking at the potential for these data together.